Welcome, everyone. Thank you for joining us tonight for what promises to be a fantastic discussion on the aging brain and an exploration of the latest and most promising research approaches to understand and ultimately treat Alzheimer's disease. As I'm sure you are all aware, millions of Americans are living with Alzheimer's or other dementias. An estimated 6.2 million Americans aged 65 and older are living with Alzheimer's in this year, 2021. This translates to more than 1 in 9 people aged 65 and older. That's over 11% of the population. And as the size of our population, age 65 and older, continues to increase, and the world population continues to increase, the number of people living with Alzheimer's or dementias will only grow. So disease-altering treatments have proven really challenging to find and develop. While we recently heard of the first Alzheimer's drug approval in nearly 20 years, we have a long way to go before we can effectively treat all patients with this disease. But hopefully, as we'll touch on tonight, perhaps we can identify those patients for whom this new treatment might actually help. So what this means, though, overall, is that we still don't fundamentally understand what is happening in this disease process. And this is the really critical step in the path towards truly curative therapies. At NYSCF, our Alzheimer's Disease Research Program was one of the first disease programs we launched because of the opportunities that stem cells give to let us have a glimpse inside the human brain, to access patients' brain cells without cutting open patients for brain biopsies, and to study those cells all without harming patients themselves. It basically is a window into a living brain. Countless studies powered by stem cells have allowed us to get closer and closer to understanding what causes this terrible disease. The main hindrance in developing treatments that actually work. We can't treat what we don't understand. Today, we're excited to discuss a landmark study performed by our panelists and collaborators that use stem cells to capture the diversity of the brain across individuals who age very differently, some healthy, some with Alzheimer's, and many in between. The remarkable thing is that stem cell models of these individuals capture this continuum beautifully. So this research really leverages the potential of human stem cells to discover what makes each of us unique, down to how we each experience aging and diseases like Alzheimer's differently. These are the insights that will lead us to precision medicine, find the right treatment for the right person at the right time. And this approach has never been as urgent as it is for Alzheimer's disease right now. The more we understand about Alzheimer's and other dementia disorders, the more we're going to have opportunities to both treat and prevent disease from happening towards the goal of healthy aging for everyone. Before we dive in, I want to acknowledge the critical support and transformative partnership of our supporter, the Stavros Niarchos Foundation, which makes events like today's talk possible. Our research and discoveries are fueled by philanthropic support, and I'm very grateful to those of you who have considered joining us in our mission to accelerate cures for all patients. So I want now to introduce Dr. Scott Noggle, NYSCF Senior Vice President of Research, who leads our Alzheimer's disease research here, to explain this latest work and kick off our discussion. Scott.
Thank you, Susan. Share screen. Can everybody sees the slides, right? Okay, great. So yeah, I wanted to start off tonight's discussion to give a little bit of background on some of the different technologies that we're going to be discussing over the coming hour, and a little bit of background on Alzheimer's disease and why we think we can use stem cells to actually help understand and tease apart what's going on in the brain of people that that are experiencing Alzheimer's disease and other age-related dementias over time. So Alzheimer's disease has really traditionally been diagnosed after death to a large degree. For more than 100 years now, we've known that in people that have Alzheimer's disease, most of those people have accumulation of different proteins in the brain. And these are the amyloid plaques, the tau tangles, and also over time, the brain degenerates. Particular areas of the brain, you start to lose neurons over time, and this leads to many, many clinical symptoms that we can discuss tonight. But this problem of only being able to really know that you're looking at Alzheimer's post-mortem or after death has been something of a hindrance in trying to understand the mechanisms of how the brain succumbs to Alzheimer's disease. So tonight we're going to talk about a different model for trying to understand what goes on in the brain, and that's using stem cells. But just to get everybody on the same background, there remains many different, understanding on how Alzheimer's actually arises. And we know that this amyloid buildup, these protein buildups can be either absent or present in some individuals that either have symptoms or don't. A lot of the therapies that have been developed have used mouse models that may only tell part of the story. And so one of the unique aspects of what we're going to discuss today is being able to use a human system to actually understand human diseases. But we know that Alzheimer's disease is a complex disease. It has many genetic factors and subtypes. There are people that have mutations in single genes that guarantee that they'll get Alzheimer's, usually very early in life. But the vast majority of people get Alzheimer's very late in life, and we know that the genetics underlying our susceptibility to Alzheimer's late in life is quite complex, and we really don't understand how these different genetic factors play together to cause disease. And Alzheimer's may not be just one disease, but actually may be many diseases. To understand this, we can't really treat what we don't understand, as Susan mentioned. But one of the unique features of the model that we're going to discuss tonight is being able to actually look at the cells that are affected by disease. Stem cells allow this window into disease, but they also allow us to capture our individual genetics. So when we create stem cell lines from people with disease, or with different diseases, we're capturing their genetics, and these stem cell lines capture the genetics in such a way that we can understand the genetic impact and have a system for teasing that apart and understanding how that leads to disease. Stem cells we call them sort of the superheroes of the body. The kind of stem cells that we study are cells that can become any cell type in the body. They can make neurons, they can make blood cells, they can make muscle, all of the different cell types in the body. And they also can be expanded. They can make a sort of an infinite renewable supply. So we have many opportunities to use these cells to study and understand how the disease process works. In the laboratory, when we make these cells, we can start from skin or blood, and using sort of a genetic trick, we can trick those adult type cells, those adult cells, into becoming these cells that we call induced pluripotent stem cells. These are cells that look and behave very much like embryonic stem cells, or the cells that you can isolate from very, very early embryos. They have all of the same characteristics of being able to make more of themselves and make all of the different cell types of the body, except that we can make them from individual people now. So we can expand these and convert these into the different cell types that are affected by disease. We can make these all the different cell types that we find in the brain. So the brain's composed of not only neurons, but these oligodendrocytes that surround the neuron and sort of insulate them, and other cell types, glial cell types in the brain that help support and clear debris from the brain, and also sort of signal sort of immune system characteristics of the brain. So these microglia that sort of patrol around and find cells that have died or other debris that needs to be cleared in the brain, and many other functions as well that we're beginning to understand. So we can make these cell types, neurons that are the communicators, oligodendrocytes, the cells that wrap the brain, wrap the neurons, astrocytes that are sort of like waste collectors. They recycle different components of chemicals that the brain uses to communicate. The microglia that go around and patrol and try to find things that shouldn't be there or things that are going wrong in the brain. And we in the laboratory can also sort of assemble these cell types into organoids, these little sort of mini brain-like tissue that allow these cells to sort of communicate and interact with each other. And we can sort of tease apart how these different cells might play a role in disease as well. So stem cells are a model or a window into different disease processes. So if we take healthy people and take people with a disease, we can create their brain cells and sort of compare them and ask, sort of, what is the first thing that we can see that is different between brain cells from people that have a disease versus those that are healthy. And that is the sort of the basis for the study that we're going to talk about tonight, and the work that we've been working on with Dr. Bennett and Dr. Tracy Young and other collaborators, starting from a very unique cohort of people that captures sort of aging and cognition across diverse individuals. And at NYSCF, we used our automation technology to create stem cell lines from this cohort, and with Tracy and others have made the cells into the different brain cell types, including neurons, and these have shown striking similarities to the brain tissue that these same individuals have shown. And we'll get into discussing sort of more of the details of what we found here. But we've set up a platform that allows us to be confident that we're seeing the changes that happen in the brain in the dish, so that we can study those and tease apart what might be happening, with the long-term goal of being able to find new and unique treatments that really address the individual characteristic of people's disease. So with that, I want to end and introduce Dr. Raeka Aiyar, who is going to moderate our discussion and drill down into more of the details of the study that we've done.
Thank you, Scott. I'm really honored to be moderating today's panel on this really exciting and important topic, and on a landmark study that was published this month by our panelists, including Scott, that we're all looking forward to delving into. I want to thank those of you who have submitted questions in advance. I'm going to be incorporating those as we go along, and invite all of you to share your questions as we go using the Q&A function in Zoom. Please do note that we're not able to answer any personal medical questions, as we're focusing today's discussion on the latest research, current treatments, and the road to better treatments and cures. So with that, I will bring on our additional panelists. I'll start with Dr. David Bennett, who is the Robert C. Borwell Professor of Neurological Sciences at Rush University and Director of the Rush Alzheimer's Disease Center. David, we're really delighted to have you here with us today. You've been sort of the architect of this fascinating ROS-map cohort that Scott just told us about, which is the basis of the study that we're going to get into. Could you tell us a little bit more about what makes this cohort unique and valuable for gaining new insights into aging and Alzheimer's?
Yeah, so these two cohorts are quite unique. The religious order study is a study of over 1,500 nuns, priests, and brothers from across the United States. And the Rush Memory and Aging Project includes more than 2,200 people across all of northeastern Illinois. And the cohorts are harmonized, and they have a couple of features that make them very unique and very different than any other studies on the planet. So first is we enroll people without dementia. and we test them every year. So there's studies that enroll people without dementia and survey them over time. A 1 year follow-up window is actually on the short side of most population or community-based studies. But the really unique feature is that a condition of getting in for all 3,700 plus people is that they must agree to organ donation at the time of death. They have to sign an anatomical gift act in order to get in the study to begin with. And the result of that is that most of them, almost all of them, when they die, we do get their brain from all of them. And then for everyone that dies and has an autopsy at the Rush Alzheimer's Disease Center, we also get spinal cord, nerve, and muscle. So these people are tested every year. We get blood every year. And then we get brain, spinal cord, nerve, and muscle. And then more recently, some other pieces of tissue, such as bone and fat. And then another unique feature is that we draw their blood every year. And we actually take a couple vials of white blood cells and we cryopreserve them. So we put them in a solution that preserves them, keeps them alive at -80 degrees in a freezer. And then we can grow those up at a later date. So if you would have asked me starting in 1994, what was I going to do with those cryopreserved white cells, I would have said, well, nothing, probably for a long time. And we put brains in the freezer every year. And if you would have asked me then, what are you going to do? I would say nothing, probably for a very long time. But I knew a couple things back then. One is that it was the beginning of the Human Genome Project and the effort to actually map the entire genome, and it was driving a lot of technology innovation. And then after the genome, all of the other molecular machinery that's of interest for cognition is in the brain. So you can't really study the brain outside the brain unless you can do one other thing, and that's where the New York Stem Cell Foundation and Tracy come in. So long after these people have died, and we've characterized the brain, and we've characterized them during life, and we've done all this molecular omics, we could then take those white blood cells that are frozen, we can send them to New York Stem Cell Foundation, they can bring them back to life, okay? And then they can convert them into baby cells that can become anything, right? And then we pass them off to Tracy, and Tracy can tickle them until they become little brain cells. And then we can study them. And so there is no other cohort on the planet that you could do this. And you could do brain cells, we could do spinal cord cells and relate them to the spinal cord. So we're talking about a way of getting to personalized medicine. I can make these cells on anyone on this call, or I can't, Scott can, all right? So we can take blood from anyone on this Zoom call, all right, and we can make them into iPSC and we can study them, and we will know something about what's going on in their brain, right?
Right.
So that's it in a nutshell. And there's no place else to go to do this. If there was, Scott would have done it. We did it. Tracy did it. You know, we did it together as a team.
Right. Thank you. I mean, that's a lot of possibilities and a really, really deeply characterized cohort. And we'll come back to a bit of that data and how we're making use of it in a moment. But now I want to introduce our final panelist, Tracy Young-Pearse, who is the Associate Professor of Neurology at Harvard Medical School, the Associate Chair of Neuroscience Research at Brigham and Women's Hospital in neurology. So Tracy, could you tell us a bit more about how you got involved in this project and what was your goal in making stem cell lines from this cohort that we're talking about today?
Sure. So we had started studying stem cells from people with early onset Alzheimer's disease, people who have a mutation in one specific gene called APP, and people with that mutation invariably develop Alzheimer's disease. So it's a fully penetrant, very strong mutation. And there, as Scott had mentioned in his intro, we could capture the genetics of those individuals and then study brain cells from those. And we were finding seeing really interesting things. Scott also was doing similar studies at NYSCF with some other familial Alzheimer's disease patients. That work gave us hope that this brain in a dish model could really help inform us about disease. And around the same time, I was introduced to David and his cohort. And what really struck me that was not only the immense amount of data that was captured from these individuals, but something that David didn't mention is what a generous collaborator he is. And so he has hundreds of collaborators around the world that he shares not only biospecimens with, but also data with. And that is so incredibly valuable. So not only does he share it all, but he also insists that they share the data and the reagents back with the world. And so one other thing that makes this cohort is so powerful is that all of this information that's acquired from hundreds of laboratories is all shared and aggregated in a way that we can all use to help us understand disease. And what I couldn't believe was that we didn't have stem cells from this amazing cohort, because it's so rich, and you have living blood cells that are cryopreserved, but wouldn't it be great to have an unlimited supply of cells from these individuals? And then even better if we can make them into any type of cell in the body. I will say that some of these cells were frozen in the freezer since the early 90s, as David said, and not necessarily for this purpose. They're meant to be kept alive, but that's a long time to be in the freezer. And I'm not sure anybody besides NYSCF could have actually brought them back to life. Because while stem cells, people can make stem cells, lots of people make stem cells from skin. NYSCF and Scott have developed through a lot of hard work and trial and error, strong pipeline to rejuvenate these cells to make them into iPS cells. And so that was the inspiration, was how struck I was by the amazing cohort and network of collaborators that David had assembled, and then the ability of Scott and his team to make these into stem cells, and we had the confidence that they would be able to overcome the challenges of however many years, 20 plus years of cryopreservation.
All right, yeah, excellent. Well, before we get into the study itself, I just want to take a quick step back and, talk a little bit more. Scott set this up for us, but, what is it that makes Alzheimer's disease such a complex and challenging disease to study and find treatments for? And, what unique advantages were you hoping to exploit of stem cells as you set out into the study in order to sort of overcome some of these challenges? Scott, do you maybe want to start?
Yeah, like I said in the introduction, we know that genetics plays a large component in your risk for developing Alzheimer's disease, but it's been very, very difficult to understand how our differing genes and genetic makeup actually, how they play out, how those genes interact and function to actually cause disease. And there's been at least 30 different genes and mutations, variations in the genome that have been described to give you some risk of Alzheimer's disease. But it's not clear how those variations actually work in the cells that are succumbing to disease. So that sort of cause and effect that we need to identify to really understand which genes and which molecular players in the cells we need to target with therapies. Trying to tease apart how the genetics impacts that has been very difficult. So because the stem cells actually capture our genetics, our differing genomes in the system, it gives us an opportunity to sort of tease apart this cause and effect relationship. If we go in and look at the cells and look at their genomes and understand how those genomes impact what we see play out in the dish, it gives us an experimental tool to sort of understand what's important and what's not, what's a cause and what's a consequence. And I think that's one of the main features, and I think Tracy and David probably have additional ideas on why Alzheimer's is so challenging to understand.
Yeah. So
Also, if you could speak up a little bit, David.
yeah. Sorry.
That's all right.
So if you think about where we've had the greatest success in personalized medicine, which is cancer, all right? If you study diseases of the brain, you usually suffer from cancer envy, all right? And it's not that you want cancer or you want anyone else to have it. It's just that it's fairly easy to study relative to brain diseases. You can take the cancer out. You can look at the molecular fingerprint of the cancer. You can give somebody a drug. You can take the cancer out a second time, and a third time, and a fourth time. And so the path to understanding the biology is much more rapid than a disease where you have to wait for people to be dead to study it. So that's the first thing. The second thing is this thing that we call Alzheimer's disease. All right, it is a disease, but your brain is full of diseases. There's at least a dozen different brain pathologies in your head. Your brain, like other systems, does not want to be demented. All right, it has ways to protect itself, and so there's numerous factors that the brain uses to protect itself. And so you've got this complex set of multiple pathologies with multiple different abilities between people to protect themselves from these pathologies. And all of that coalesces as a syndrome that is a progressive loss of episodic memory, the autobiography of your life, all right, that eventually results in a dementia, right? And we call that Alzheimer's disease clinically. But when you get under the hood in the brain, Alzheimer's disease is a very particular type of brain pathology, all right, in a sea of other brain pathologies, all right? And you cannot separate those out in living people. You can see a couple of the pathologies, but you can't see all of them, and you can't see the resilience factors that are there. And most people that have Alzheimer's disease Alzheimer's dementia, have more than one pathology. And in fact, if you only have Alzheimer's disease in your head and no other pathology, the chances are you don't have dementia, because it's really the multiple hits that get you over the edge. So we can identify these people while they're alive, but we really don't know all the other things that are coexisting with the Alzheimer's pathology. So that gets us to some type of model system, okay? And the common model system is a mouse. It's a mouse with a human gene that causes Alzheimer's disease in relatively young people. And sometimes it's 2 of those, or 3 of those, or 4 of those. And it bears little relationship to anything that happens on the planet naturally. So we have given and cured Alzheimer's disease in mice hundreds of times, right? And mice don't get Alzheimer's disease, right? So there's very few species on the planet that get anything like this, right? It's in parts of the brain that exist in mice, but it's in other parts of the brain that don't exist in other non-primate species. All right, so it's an incredibly challenging disease. It may not be the most challenging disease. Okay, the black hole that some people go into is schizophrenia, which is a younger problem. Again, no biomarkers, and then they die in old age. All right, with all these pathologies as well. So I wouldn't even try and tackle that. But here, and I'll let Tracy talk about it, but here's an opportunity to take advantage of the fact that, Scott talked about the individual genes, but if we just talk about all of the genes together, and we take something called a polygenic risk, right? So all of the genes together, right? The genetic effect of driving amyloid and tau, which are the two pathologies of Alzheimer's disease, is actually pretty strong. And we're really leveraging that strong genetic component in these iPSC. If you don't have a strong genetic component, you can't use these baby cells. All right. But if you do have a strong genetic component, all right, then basically we are doing something different than what is usually done with iPSC. So Tracy talked about you get an iPSC from somebody with a mutation, and you do a whole bunch of experiments on that one line with that one genetic background. So here we're basically saying we're going to do something, we're going to marry stem cells with epidemiology. So we're going to look at person-specific differences in the living human, in the autopsy brain, okay, in the pathology in the brain, in the molecular genomics in the brain. Okay, and then we're going to look at those same person-specific differences in the cells in a dish.
Right. So what a great opportunity. Let's get into the study, Tracy. I think, since you led this study, your team published this in the journal Neuron on early September. And I think we'll have that pasted into the chat in a moment. So can you talk to us about, what are the main findings from your study? What did you learn about Alzheimer's that was previously unknown? And did you find any surprises that you didn't expect?
Yeah. I mean, first of all, yes, there were lots of interesting things, and I'll try to hit this high notes. One thing that I was very struck by and surprised by right away is that when we made neurons from these 50 different people, and we measured the amyloid that the neurons made. So Scott talked about amyloid accumulating in the brain of Alzheimer's disease subjects invariably. And this amyloid, though, comes in many different forms, different lengths and different modifications. And the neurons of the brain really make the vast majority of the A beta, which is also called, another word for amyloid is A beta. And what we found was we measured all these different types of A beta, and I was really surprised and struck by the fact that people with different genetic backgrounds, and these 50 different people, made different profiles of A beta. And the people with Alzheimer's disease, their profile of A beta as a population was slightly different than the people who didn't develop Alzheimer's. And so really it wasn't necessarily the total amount of A beta produced that was different in people with high amyloid in their brain versus low amyloid. It was the specific subtypes of A beta. And so we think that, as Scott mentioned, we're capturing the genetics of these individuals and the genetic risk for Alzheimer's, this polygenic risk, as David described. And we think that many different variants in the genome are contributing to how these cells are making A beta and the type of A beta they're making. And that was surprising. We didn't necessarily think that we would be able to observe that. And what that resulted in was we could actually see a significant association between what we were measuring in the neurons in a dish across these 50 people and what we could see in the brain of those same 50 people. Both the amount of A beta plaques that were present in the brain, but more importantly, with their trajectory of cognition. So their cognitive scores at death and the trajectory of cognition over the last decade or so of life.
Right, because they all started out normal, right?
Normal, exactly. And then because David has this nice longitudinal longitudinal cognitive testing until death, we could look at the trajectory, which, some people in the cohort had very stable cognition out to 100 years old. Some people had Alzheimer's and very steep, and some people had Alzheimer's, but it was a relatively slow progression. And so what was really surprising is what we could measure in the dish had some predictive value in what was observed in terms of cognition and pathology in the brain. So that was really surprising. And then beyond that, we also did a lot of unbiased analyses of the neurons to identify networks of genes and pathways that were also associated with polygenic risk for Alzheimer's and with the development of the disease. And there, we also had a number of different pathways that were highlighted that are stimulating many more additional studies going forward. And if there's time, I'll get into some of those, but I'll stop there so that we can get more questions in.
Thank you. Well, that's great. And thank you, David, as well, for that really thorough explanation of sort of what the challenges are that we're taking on. Scott, is there anything you'd like to add that you think was surprising or unexpected for you as you guys looked at the results of the study?
Yeah, and I think Tracy can probably talk about this a little bit later, but this ability to use the cells to sort of study cause and effect, and there's been sort of a long-standing idea or sort of hypothesis and lots of evidence that there's some sort of connection between the accumulation of different amyloid A beta species, and these are like proteins that are usually excreted and sort of in the between cells in the brain. They build up into plaques that that you can see using various methods. But there might be a relationship between those and the tangles that develop inside the neurons of the brain. And sort of that cause, that link has been sort of hinted at in a lot of different studies, but Tracy can go to the details. But we've found sort of an interesting molecular chain of events that we think might lead from one to the next, and I think that really sort of exemplifies a unique feature of being able to have this brain-like tissue in the laboratory to study and sort of understand. You replay the movie over and over again. Can you find sort of causal links in this chain that might explain, how pathology develops, and that then leads you to where you need to actually poke and prod and find drugs that it might actually, alter that disease process. So for me, the demonstration of this ability to show cause and effect in the dish that has relevance to the actual disease in the brain was very, very exciting for me.
Yeah, that is really exciting. And you touched on an important point.
Raeka,
please.
I just have to add something because
Yeah.
I too was surprised. Okay, and I have a higher level of surprise, which is that this worked at all. All right. I mean, let's face it. I mean, while we were doing this, our colleagues around the country were telling us, "Oh, this doesn't work because they're baby cells in Alzheimer's disease. It's sporadic disease. It's a disease of old people.
Right.
These are people that died at age 90. Okay. When you make an iPSC, you are making a cell, okay, that is a newborn brain cell. So this isn't a baby cell in the sense of a 3-year-old or a 3-day-old. Okay, it's a baby cell in the sense of a newborn neuron that occurs very early in the embryonic stage. All right, long before birth. All right, and while we were doing this, our colleagues are telling us this isn't going to work. All right, and so that it worked at all is amazing. All right, and that it worked in 53 people. Realize we have over 3,700 people in these studies. We have over 1,700 autopsies. We publish studies with 1,000, 1,200, 1,500, 3,500 people routinely. And here we got 53 things that never should have worked, that worked really, I mean, astonishingly. And so, we did the work. We didn't, okay, but I think people need to really think about what it is that is here. And again, because everyone on the Zoom call, we could get their blood, we could make these cells, and we could test them. So it is finally a bridge, right, to being able to do something in living people based on what we've learned in the dead people. All right.
Absolutely.
And for somebody who's been doing these cohorts now for over 25 years, I want to see that bridge. I can't help these dead people anymore, but I can help their children and their grandchildren, and my children and some future grandchildren.
Well, I think we're all really glad that you went for it, despite the odds and the critiques. It's really exciting to see. And do you think, to your point about these cells being very young, there's an estimated, I think, 2 or 3 times the number of people who currently have Alzheimer's who are thought to be pre-symptomatic, who are going to develop it. Do you think that that means we'll be able to identify those people before they develop the pathology and intervene in that?
I think it's possible. It creates a possibility.
Yeah.
We're not there yet, but it creates the possibility. It'll require a lot more cell lines, a lot more nuanced understanding of the genetics lined up with the different types of species, that Tracy is assaying. But it makes all of that possible. And even getting the money to make the iPSC, wasn't easy. People want to know, why do you need so many cell lines? Because they're so used to studies of one cell line. Like, why would you want to look at the genomics, right? Backgrounds. We're like, well, that's what makes us different. Not just the genomics, but all kinds of other things that happen to us during life. So if we're going to get to this thing called, quote unquote, personalized medicine, you got to get down to the person.
That's right. Absolutely.
Different people, not just one person.
And so the causes can be different in different people, right? You were talking about causes, Scott. So, we hear that it's, Alzheimer's is caused by a combination of genetics and environment, aging, other factors. So
I just want to say that Alzheimer's disease is this amyloid in tau.
Right.
Alzheimer's dementia is what has multiple causes.
Right.
And of course, the amyloid and the tau also have multiple causes.
Yeah, I was going to say that, David.
Okay, but there is a pathology that is Alzheimer's disease that the field in general has agreed upon based on the recent workshop by the National Institute on Aging and the Alzheimer's Association put out a paper on that. Okay, so the disease is complicated, but there's all kinds of other things that make it more complicated to be studying in the living human.
Right. Well, thank you for clarifying that. I think, from what are the sort of most significant factors that we know about? We've had this question from the audience in terms of what is causing, this amyloid and dementia type symptoms. And are there opportunities in your data or with these stem cell models to better nail down these root causes across different individuals?
So that's a really complicated question. The data set itself is designed that the autopsies is really about things, but one of the things is we look at risk factors for clinical disease, and a major goal of the study is to understand, well, how do those risk factors impact the different types of brain pathology? Okay, so it turns out that most risk factors for Alzheimer's dementia, we have published on many of them. So lifestyle factors, medical factors, genetic factors, personality characteristics. We've identified numerous things that cause memory loss, Alzheimer's dementia, and almost none of them cause this amyloid in tau. Okay. One is surgical menopause in women. The women that have their ovaries removed before menopause, right? That's associated with memory loss. It is associated with this AD pathology in the brain. But almost nothing else has a direct association other than the genetics.
And that's what we were capturing in the neurons, with genetics, and that's where we saw a difference in A beta. But as Scott mentioned, there's this, one of the interesting parts of the study was this connection that we identified between the A beta and the tau. And there were different pathways that seemed to be linking the two. And one of them was this protein phosphatase that was induced by, it was reduced in expression. So it was not as active when you had this bad A beta, Alzheimer's A beta, and that resulted in a worsening of the tau. But what was interesting in the study is that it wasn't in every single Alzheimer's disease subject. So some Alzheimer's disease subjects responded to that change in A beta by lowering this enzyme, but others were all protected from it for some reason, and most likely genetically encoded. And so this kind of speaks to the power of these cells in capturing the genetics and these subtle differences across humans, but it also opens the door for personalized medicine, as we talked about before. This is an example where if we think that affecting this enzyme could be a therapeutic option to pursue in Alzheimer's disease, we might only want to look in those humans who responded to the A beta in the way that altered this enzyme. And that's just one example, but there are many similar examples from using these, in terms of using these cells when they become glial cells or other cell types in the brain, where they may have deficits in a subset of patients, and then you might need a different type of intervention. So not only do these cells provide a way to identify the different causes, the different roads to Alzheimer's dementia, but they also allow a platform for testing that hypothesis and then testing drugs that could help those individuals.
So that's a really, really valuable resource of both the cell lines and the data. That's, it seems like it's going to enable a lot of things going forward. How else is this resource that you all have created being used, and how would you like to see it used and built on going forward? Like for example, I mean, one of the major challenges with these cohorts is ensuring diverse representation. This is really important given the higher mortality rates that we see in ethnic minorities for neurodegenerative diseases like Alzheimer's. So how are you thinking about this going forward, building upon this work and making sure it's maximally useful?
All right, I'm going to try and talk louder. Better?
Yes, that's great.
That's a really important question. There are some genomic differences between people of African ancestry and Native ancestry in terms of developing Alzheimer's disease. An obvious example is something called ApoE4, which in Europeans is the most potent genetic risk factor for Alzheimer's disease. It's much more common in people with African ancestry, but it's much weaker. So it's much more frequent, but it's much weaker, suggesting that it's under what's called positive selection, positive natural selection within Africa. So it has some protective effect in people with African ancestry. So we are now funded to generate 57 cell lines from African Americans, all right, where we'll be able to look at their polygenic risk and then be able to see whether we can replicate what Tracy did in the African Americans and how that's different. To bring in genetic diversity with ancestry is going to be a lot more complicated because the vast majority of African Americans are some combination of African ancestry and European ancestry, sometimes with mixed in Native ancestry. And so that's all roaming around the genome at the same time. And so you can have a piece from Africa and a piece from Europe, followed by a piece from Africa. And you can think of it as a 3 billion base pair deck of cards, which has only been shuffled 20 times. All right, so it's not surprising to find long runs of cards that are in order, all of one ancestry. and then opt-in is some other ancestry. So very common. Yeah, well, maybe 22 times depending. But you're looking at, you know, I'm sorry, somebody wrote a comment that I'm responding to. Yeah, about 400 years.
David, that's why we would love to make lines for more people, right? So that we can
Yes,
have
yeah.
more of the genetic. And I think David and Scott and I are all on board for that. So hopefully with this next 57, we can convince our funders that we need more, that we can do it and we can find interesting things, but that we could find even more with more numbers.
Right. And another thing people are interested in, and they've contacted me, and I suspect some have contacted Scott and some have contacted Tracy, is do you have lines with particular genes or particular combinations of genes that I want to study? All right. And so being able to support that kind of work across a wide range of people and interests is important simply for speed.
Yeah.
All right.
Right.
Speed meaning we, the four of us, and most of the people on this Zoom call, all right, are looking at Alzheimer's disease in our future because we're going to get old. All right. And so there's that target timeline, time horizon, and then of course there's the next generation timeline. So so one of the reasons that we're so committed to an open science in sharing everything widely is simply that, you know, the clock is running on all of us. All right. And so the sooner we get this done, you know, the more good it's going to do for the greatest number of people.
And actually that, David, reminds me, Raeka, one of the things you asked about was what else these cells are being used for. And it's a huge array already. And the word's just getting out. So there are people studying susceptibility to viral infections and how that affects dementia. People are studying diabetes connections with Alzheimer's. Of course, Alzheimer's mechanisms in many different cell types, including the blood-brain barrier, astrocytes, microglia. It's really amazing what I think. Hopefully the data will all be shared from all of these different studies, but I think we're going to see a lot of good payoff from generating these lines.
And I just reiterate what David said, that the more eyes you have exploring the problem, the faster we can go. And so getting this resource out to the greatest number of people should speed things along, hopefully.
Yeah, absolutely. So your data so far tells, you know, a couple of different stories. It tells a story of this disease being a lot more complex than we think it is. It tells the story of a sort of continuum of aging, that you see, you know, so much variation in individuals and the way that our brains age, and all of these pathologies I'm trying not to think about that we seem to have in our brains that David mentioned earlier. But that points to the opportunity that we've also, you know, hinted at of you know, sort of tailoring treatments towards these different categories of aging. So, you know, how do you think going forward you would like to, like, what do you see as our greatest potential for being able to discover these kinds of tailored treatments and, you know, precision medicine type approaches for Alzheimer's disease? And could we, you know, we've just got a few minutes left here, so I'll combine that with another question that we've gotten, which is, you know, could we foresee a path to healthy aging that maintains healthy cognition for everybody? You know, would there be some intervention strategies that look promising or might be on the horizon? We get, you know, questions about dietary strategies, other medical interventions, et cetera. So Tracy, why don't we start with you, and then we'll.
Sure, I'll kind of take that. I'm going to take a narrow view to that question, just because I wanted to bring up Aduhelm, which is something that people have heard about in the news.
Or aducanumab, as others might have heard of.
Aducanumab, yeah. And it's an anti-A-beta therapy that's been approved by the FDA that's highly controversial.
Right.
And one of the reasons perhaps that it's highly controversial is that the results from the clinical trial were mixed. And one of the ways I would like to immediately use these cells is to test whether the type of I mentioned before, the different humans made different types of profiles of A-beta. And what I would predict is that Aduhelm would engage the A-beta in a subset of people, and that might give us some clues as to how to make Aduhelm more effective in treatment in humans. And so that's a more perhaps immediate use of these cells where this drug is already approved. Not everyone seems to respond to it the same way.
Right.
And perhaps that's something we could do right away to try to take advantage of this cell collection. But there are many other things to say in answer to your question, but I'll let Scott and David.
Yeah, just testing it directly in the dish to see like how do
Exactly.
these neurons react? Yeah, Scott.
Yeah, no, and just to add to that, you know, this aducanumab story, it might, you know, affect one subset of people, but we also need to understand what the other groups are doing, and how we might be able to, you know, design therapies directed towards different subgroups. We know that, you know, when we look at brains and look at their gene expression, that you can find groups of people that fit into different buckets, and those different buckets are maybe 5, 6, 7, 8 different buckets, and those different buckets usually sort of center around different types of cells in the brain, or things that you would expect to find in different types of cells in the brain. So one of the things that I really am anxious to sort of use this for is to try to understand and see if we can predict who fits in those different buckets. And if you can understand what's going on in those different buckets, we might be able to, you know, find tailored therapies, but also exclude those people from going into clinical trials in the first place if they're not going to be reactive to the drug that's tested. So this sort of dual, you know, making clinical trials more efficient, you know, better able to be predictive, I think is one of the, you know, I think one of the more exciting possibilities for using this resource.
Yeah, especially when you think of how many billions and decades go into getting a successful drug to market, which we won't go into. David, we'll give you the final word on this.
Yeah, so I mean, I think, you know, when I kind of look into the crystal ball as to potentially what can be done with this, it's basically doing different compound screening for its effect on the development of these pathologies in a dish.
Compounds, you mean drugs?
Drugs,
Yeah.
or things that could be drugs, and then figuring out what genomic backgrounds seem to be responding to this drug. And then you need to select a whole nother set of lines that have just that genomic background and see whether you can enrich the response. All right, and you do this in an iterative fashion. It's going to require hundreds, if not, you know, thousands of cell lines, all right, to be able to do this repeatedly in order to get to the point where we could say this genomic background, right, does best with this particular drug, right? And then, you know, for $30, $40, you can get that genomic background in a living human, all right? So to make that leap from the cells to the human, all right, to do your clinical trial in the humans that way. And so you get this crosstalk between what you can do in the dish and then what you can do in living people. And right now we think that this paper says this is a possibility for this pathology. We haven't shown anything for any other pathology. The genetic risk for many pathologies is going to be too weak, but there are other things that have a strong genomic driver to it. And so we may be able to do this with other diseases as well. I think it's one of the most exciting things that has come out of literally over 25 years of work, all right, with over 1,000 manuscripts that have been published from this data set by us and by people around the world. So
wow.
So it's not that it's, I mean, it's really widely used, and I just want to be able to get to the point before I retire that we have helped bring something to market that actually helps people clinically. I'm a neurologist, I take care of these patients. And I've been doing it for 3 decades. And it's just a terrible, terrible disease to watch.
Yeah.
Just terrible.
Wow.
And I will say that, you know, we tend not to think of it this way, but this disease kills you. It doesn't do it in months. It does it in years. All right. But there's no market for a T-shirt that says "I survived Alzheimer's disease." All right. And so Alzheimer's disease is in a horse race with death. For most of human history, death won. Now we've succeeded in medicine at putting death off. So Alzheimer's disease comes first in too many people.
Well, thank you for that, David. And I really appreciate, I think I speak on behalf of all of us when I say appreciate the 3 of you bringing your teams and many other teams together to take this really innovative and open science approach to finding out what's going on in this disease and, you know, paving the way towards better treatments. I definitely feel more hopeful after hearing this conversation. I do apologize we couldn't get to every question that we received, but do thank you all for submitting those questions. There were questions about being able to access the cell lines. Please do reach out to NYSCF. We'll send a follow-up email before that you're welcome to reply to. And also, if you'd like to get involved with our any of our clinical research, we're always looking for participants. That information will go into the chat and into our follow-up email tomorrow. And if people are interested in supporting making additional stem cell lines from this cohort, as you know, Tracy and all of you underscored the value of expanding this cohort. You know, we would certainly welcome any support we can get for that. So please also do get in touch. So I want to thank you all again for a fascinating discussion, and we hope to see you at one of NYSCF's future events. And until then, keep up the good fight. Thank you all.